So today our topic of discussion is glycolysis, and from glycolysis I mean breakdown of glucose into pyruvate molecule. So if we look at the general structure of glucose, it's a circular structure and it's a cyclic structure basically, and it contains 6 carbons. If we number those carbons, there are 1, 2, 3, 4, and 5, and 6. So we need to remember these positions because when we're discussing the glycolysis, these positions are going to be very effective in naming the whole process and the whole reactions. There are hydroxyl groups attached at the certain places and certain position of the carbon, and the whole formula of glucose is C6H12 and O6. So basically, glucose is the form of energy. It's a carbohydrate that we take inside the body to gain energy. So in order to glycolysis to take place, this glucose needs to get inside the cell because glycolysis is going on in the cytoplasm. And this glucose molecule is present extracellularly. So there's extracellular space and this is intracellular space, and this can be differentiated by a phospholipid bilayer. Now this glucose molecule needs to get in intracellular space so that it can just start the process of glycolysis. So how will it do that? Well, there are certain receptors that it's going to be attached with, and then it's going to go inside our cells, and these receptors are known as glut receptors. These are the receptors and through which it's going to be passed inside the cell. There are 4 types of glut receptors that helps in this molecule transfer, this glucose transfer inside the cell. They are namely GLUT1, GLUT2, GLUT3, and GLUT4. There's also one more receptor known as GLUT5, but it aids in the communication in the transport of fructose molecule. So we're not going to discuss that here. And if we talk about these 4 receptors, they're just basically classified according to the location, according to where they're present. So GLUT1 is present in, you can just remember them by a mnemonic known as BBB. And from 1B, it stands for blood, like it's present in endothelia. B for baby, like it's widespread in fetus, and blood-brain barrier as well. And if we talk about GLUT2 receptors, they are present in kidney, liver, and pancreas. And if we talk about GLUT3 receptors, they're basically present in neurons, placenta, and kidney. And GLUT4 receptors are mainly present in muscles and fats, and if we technically name them, adipose tissues. So if we talk about these first 3 receptors, GLUT1, GLUT2, and GLUT3, all 3 of them are insulin independent. They're not going to be influenced by insulin. However, GLUT4 is influenced by insulin, and its expression is proportional to insulin levels. This process is known as facilitated diffusion, through which these glucose molecules are going to enter inside the cell. So that the process is known as facilitated diffusion. Alright, so now we have established that glucose has entered inside the cell. But what if there is a higher concentration of glucose inside the cell? So glucose would like to go outside the cell, and for that it's going to go follow the receptors known as SGLT, sodium glucose transporters and symports. They're also known as that. And the process that they're going to follow is secondary active transport. When there is too much glucose inside intracellular space, there is going to be a process known as secondary active transport, and the receptors that are going to act are SGLT, and they're going to move this glucose outside the cell. So now you know how this glucose is going to enter inside the cell and how it's going to get outside the cell. So now in the next section, we're going to discuss about the metabolic pathway and the whole process of glycolysis in detail. So stick around for that.